Where did we leave off yesterday? It's different classes. I think we left off at different places. Let me check the notes. Hold on. So again, as a reminder that these organelles are something you should have made flipboards, cards on. They were told about a month ago, almost more than a month and a half ago, actually, to start to know your Chapter 7 organelles, know what they are, what they look like, and what they do. We can go look at the due date of the flipboard cards, the flip cards, but I'm sure it's about a month and a half ago. One of those organelles was a cell wall. It's a cell wall is made of cellulose. It says here it's a support structure for the cell, for plants. But it's not just plants. As far as eukaryotes go, most of them are going to be plants. But let me just make a side chart here and say eukaryote. And then let me clarify that because that looks a little messy. Eukaryote. And then there's another term, prokaryotes. And where you're going to run into problems, I think, a lot of times is when words are very close together. And I keep mentioning that because I see that over and over again as mistakes that kids make. So obviously, at least at this point, obviously, we have two kinds of eukaryotes. We have the single-celled eukaryotes. Those are organisms that only have one cell. And then there's multicellular eukaryotes. Of single-celled eukaryotes, you have two kinds. Let me erase this again, just trying to keep it all neat for you. Single-celled eukaryotes, you have two kinds. There's protists. Look at the spelling, please. And you know how I am about spelling, so if I'm mentioning it, you should maybe make sure you pay attention. So there's teas versus oats. Okay? Teas versus oats. There's a big difference there. These guys are a whole group of organisms, so are these guys, a whole different group of organisms. They're very different. They're both single-celled, but protists tend to be single-celled. Some are multicellular, some aren't. So some protists are single-celled and some protists are multicellular. So there's a big group, protists. They are things, an example of protists, I'm going to go ahead and make kind of a and I showed you in class, let me, again with the neatness. It's a eukaryote, and we'll go over what a eukaryote is just to make sure we're all clear. Has this really interesting organelle called this contractile vacuole. They're very hard to classify. And because of that, I don't think you're going to see it on any major exam. However, you should know they exist. They're important organisms. They do a lot. They can cause disease. They're eukaryotes. So examples are amoebas that I've showed you in class. There's ciliates. So ciliates are single-celled organisms that have cilia that kind of row their ways through the water. Amoebas kind of ooze their way through water. Some of these, of course, this is again a eukaryote. Some of these are photosynthetic. They do photosynthesis. some are not. So it's a really big group under this big group called protists. All right, with an IST. All right, not an OT. So protists can be multicellular, or they can be single cell. So what are some examples of single-celled eukaryotes? Well, there's yeast. Yeast, right? There's protists, as I said, things like amoebas. A group called ciliates. Ciliates are actually a large group that have a lot of different organisms, species in it. Again, with the neatness, I'm going to get rid of some of this here. So that's single-celled eukaryotes. Now, there are some other examples of that. Might be mold. Mold are also eukaryotes, mold/fungi. So mold are fungi, all right? Yeast are fungi. So they're actually all in the same kind of group, and we're going to look at those groups today a little bit, and we'll come back to them later. Ciliates and amoebas are both protists. All right, but mold, fungi, and yeast are not. They're all single-celled. They're all eukaryotes. Multicellular eukaryotes there are some protists that form multicellular organisms. So some that, and certainly during some of their life stages they can. But some more examples of eukaryotes that are multicellular would be animals and plants. So these are all eukaryotes. Prokaryotes, there's only really two, and honestly, you could not tell the difference if you were looking under a microscope between the two. It would be bacteria and archaebacteria. So this is an ancient group of bacteria. Oh, I forgot the R. Sorry about that. Yikes. So these prokaryotes are a whole different group. And we can go look at the evolutionary chain of all of it, but let's just go on and move on and talk about what is this karyote. See, this thing called karyote. Does anybody know? There should be a lot of people that know at this point, but my guess is there are some people that you just don't know. Anybody here know what karyote would mean? Karyote? And obviously what maybe what pro means versus you. Anything? Anybody that read the chapter, made index cards, paid attention when we talked about it before? Nothing? Okay. So karyote means nucleus. Karyote, nucleus. All right, it's a root word if you like. Pro. What do you think pro means? Nothing? It means before. Like pre, all right? And you means it has it. So let me ask you, what do you think prokaryotes are? Huh? What characteristics do eukaryotes, let's do a Venn diagram real quick. I think that might help you a little bit. A lot of silence. Eukaryotes and prokaryotes. What do they have in common? Well, they both have DNA. They both have RNA. They both have lipids. They both have proteins. They both have carbohydrates. They both have membranes, cell membrane. Let me write smaller. They both have cell membranes. They both have DNA, RNA, lipids, proteins, and carbohydrates. So what's the difference between the two, considering the root words I just told you? Anybody? I like that. That's true. But considering what I just told you about prokaryotes and eukaryotes, look at this. Prokaryotes don't have a nucleus. Thank you. No nucleus. So bacteria have no nucleus. No membrane bound organelles. Does it have a membrane, a cell membrane? Yes, it does. Because if it didn't, it wouldn't have. If it didn't, it wouldn't be a cell. So it has to have a membrane, cell membrane. The other eukaryotes do have a nucleus. And eukaryotes have membrane-bound organelles. Now you have to know this material. You must know the difference. You have to know the difference. Again, what are prokaryotes? There's only two kinds. Only two kinds. Again, karyote, not protist, but prokaryote has only two kinds. You have bacteria and archaebacteria. They do a lot of different biochemistry, but the bottom line is that that's where you're at. All right? We're going to talk a little bit about a lot of different ones, but are we good with this? Let me know so I can go on. I don't want to leave anybody behind. Is that a good way of organizing the information for you? Do you see it clearly? Did you read the chapter? Are you on your texting on your computer or something? Is that why you're smiling? Why are you smiling? Are you just smiling because you're happy? You're just happy? That's nice. You're not happy. I don't smile when I'm not happy. All right. Well, are we good? Okay. So this is the kind of stuff that you would do if I was told, read chapter 7, make index cards of vocabulary words, know your vocabulary, know your cell organelles. This is the kind of thing, guys, I keep saying it, and I know you don't believe me, and I know because I've been doing this a while, and I know a lot of kids like you that have gone on to college, and they go, man, I should have believed you, Mr. Mendoza, you were right. And I'm like, yeah, I know. And I'm not being arrogant. I just know it's true. What I'm saying is true. If you don't start learning these skills, you're going to be in college in a year, in 2 years. Some of you will be in college in 1 or 2 years. Some of you are asking for letters of recommendations to go this summer, right, to Andover and go to college, right? You have to be able to process this information like this. This is the expectation. Can you take information from a chapter, from a book, from a case study, and break it down, organize it? Like this was all described to you in the chapter. You probably read through it and moved on, right? Didn't think twice on it. I understand what you did because I used to do the same thing when I was your age. But you can't do that. You're no longer a child. You're almost an adult. Okay? So you have to take this information, you have to break it down, organize it, and make sure you know the important parts. What's important here? Gosh, I'm telling you pretty clearly, know the difference between prokaryote and eukaryote. Know that difference. Know which organisms are prokaryotes and which organisms are eukaryotes. Know that pro means before a nucleus, without a nucleus, and eu means it has a nucleus. Protist isn't important to know. I would know what a protist is just because they really do have this thing called a contractile vacuole, which I showed you in class. A movie on it, I'll show you again if you'd like. But the contractile vacuole helps osmoregulate the cell. What it does is it kicks out extra water. It just pumps water out. And I'll show you how that works in just a bit. So we all got this so far? Yes or no? Yes? 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 No? There's some shaking of heads. Somebody said yes. Good. So let's go on and look at a comparison between two really important cells that a lot of people you're probably going to get. I'm going to make them two different colors. And I know for sure you had it in your chapter, a big diagram, 2-pager. It went across 2 pages and really beautiful drawings of animal and plant cells. One of the things I said, man, you better know this because this is going to be important. Mm-hmm. So you got a cell membrane, you have a cell wall. Remember, this is a phospholipid bilayer. When I draw this, what I'm drawing is this, correct? Yes? Nobody in here looks at that line and just thinks it's a line. Everybody in here sees a glycerol molecule with 2 fatty acid chains sticking off each side with a protein stuck in the middle. Maybe it's a channel protein. Everybody knows that this is a 3-dimensional object, not a 2-dimensional one like the one I have to draw here. Everybody knows that this protein goes all the way through the center. So if you were to take a top-down look, you would see something like that. Everyone here knows that when we're looking at this cell, these things move around. It's a fluid mosaic model. What the book talks about is something called fluid mosaic. Do you know what a mosaic is? Do you know what a mosaic is? Yes or no? Please help. No. Silly, and I pushed the wrong button. Okay, so a mosaic is when you have a bunch of little pieces that you put together to form a picture, right? Like this is tiles. These are a mosaic usually is made of tiles. So different colored tiles, you see that they're little squares, right? And these little tiles, you can make these beautiful pictures with these little tiles, and you'll see really fancy bathrooms with the mosaics in the wall or on the floor. Old Roman bathhouses would have mosaics and that kind of thing. Churches, some churches have mosaics, various religious people and items made image of them on the wall. It's just literally for the wall and the floor. And it's it is kind of a painting, but it's not a painting. It's made of little tiles, little pieces that come together to form a big picture. The cell membrane is the same. It has a bunch of proteins. Some of them are carbohydrates. Some of them are proteins. Some of them are lipids. But they come together to form this big picture. It's complicated. This is complicated, isn't it? It's like 5 or 6 different colored tiles in different shapes in different places. The difference between this and a cell is what? What's the difference between this and a cell? That's right. This is static. It's not going to move. All right. If this moves, I'm leaving the house. I'm running out screaming, right? Because this should not be moving. This is made of tiles and it's, cemented on the wall. Fluid, the membrane's moving all the time. So remember, everything's moving all the time. Remember that. Everything's moving all the time. So these molecules are moving. Just this channel's here right now. Later it'll be here. A little later it'll be over here. Just moving, just fluidly moving, randomly moving around in this 3-dimensional sphere, right? Yes? Okay. You have to imagine that. I can't make you imagine it, but it's there. So that's going on in the membrane. It's very active, very active. And the other thing that you should think about is that the membrane, again, I'm going to draw a line, but I'm going to draw 2 lines, two blue lines, hydrophilic heads. I'm going to fill in the middle with some yellow for its hydrophobic nature. So when we're looking at this cell membrane, there's on the membrane, internally, there are these little proteins and carbohydrates that stick out that form a matrix. There's these microtubules, they're called microtubules. They're used to transport vesicles. And they go all throughout and we call this matrix a cytoskeleton. It's like the millionth use of the prefix cyto. What does cyto mean? Everyone should know it, but of course most of us don't, because most of us don't pay attention. It means cell. Cyto means cell. You see the prefix cyto, you should know it means cell. So cytoskeleton is the skeleton of the cell, and it's made of microtubules or microfilaments, and they just move in every direction. And so the cell is not. I'm going to go ahead and draw it in this example of the and this is every cell. I'm going to draw it in here, but I'm going to then you know what I can do? I can copy it. I'm just going to draw the cytoskeleton because it would just get too busy if I drew the cytoskeleton and then everything else. But I want to make sure I emphasize that the cytoskeleton goes throughout the entire cell, and it connects. It's constantly changing. The cell is constantly making connections and breaking them, and it gives the cell a shape. It's a skeleton. Your skeleton gives you a shape, right? What would you be without your skeleton? A blob of meat, right? So without your skeleton, there would be no shape. So this cell has a shape because of the cytoskeleton. It's not bone. It's made of little cables. Think of it like a cable, very fluid cable network. Now I'm not going to keep that drawing and keep adding things to it because you can see it would get really complicated, right? So I'm just going to put that off to the side. So the cytoskeleton is a organelle. That's an organelle. You need to know that. If you did not make an index card, a study card for cytoskeleton, maybe you should. There, obviously, the cell membrane is an organelle. It's constantly changing what's in it, where the stuff is in it. That's why we call it fluid. And there are many different things in it. That's why we call it a mosaic. So it's fluid mosaic. On the outside, this is another term for outside, is something called extracellular. Extra, not meaning too much or more, but meaning outside of. Are we clear? Extracellular. You also have a matrix. That matrix is made of different things. It could be a lipid with a glucose molecule on it. It could be it could be a bunch of glucose molecules all attached together in a chain. It could be a protein attached to it, so amino acids in a chain attached to it, a glycoprotein, a glycolipid. They're all out there on the outside. So on the outside of the cell, you have this extracellular matrix, and that's what we call it, an extracellular matrix. We only reason that they call it that rather than a skeleton because the skeleton's inside, isn't it? It's not an exoskeleton. It's not made for support, but it does have a matrix that kind of sticks out. So the cell, they're attached to they're attached to the membrane, they're part of the membrane, but they stick out from the membrane. And maybe they're glycolipids, or like a lipid with a bunch of sugar, carbohydrates like glucose attached to it. There's a reason I'm mentioning this, because it's an important reason, so pay attention, please. So you have these glycolipids out here, and they go off and extend in every direction. And that goes all the way around the cell. I'm not going to continue to draw it, but you get the idea here. Does this make sense? Is this confusing to anybody? Is this confusing to anybody? Can you see how this can make a real clear fingerprint? Have you ever looked at your fingerprint? Have you ever looked at a fingerprint? It's a complicated bunch of lines, isn't it? Yeah. Lipids, they could be, usually they're lipids, so that's a really great question to ask. They can, not that they always do. Phospholipids are what make up the membrane. Remember the difference between a phospholipid and a lipid is a phospholipid has, this is a fantastic question, so please pay attention. A phospholipid has a phosphate and then two fatty acids. But then in the middle of that, you could have this giant lipid that sits in here, just a bunch of carbons, and it could just keep going, right? And then it could branch off. And then you could go ahead and add, maybe it has a big ring of lipid-like molecules, steroid or something, like a cholesterol molecule or something like that in the lipid, the phospholipid bilayer. But do you see this? These are phospholipid, and then embedded in that is this long chain of carbons. And at the end of the long chain of carbon, you could have a glucose molecule. Does that make sense? Okay, excellent. So it could be attached to it. Now, do you see how they can make a bunch of different shapes? Can you see how crazy that could get? Just like a fingerprint, right? Yes? Well, that is how your body identifies who you are. That is one example of this kind of structure. This side of what this extracellular matrix does is one thing. It does is it. Let me erase this. I don't know why I can't get any bigger, but. One thing that it does is identifies you. So this identifies you. to you. If I took and I put my liver in your body, what would happen? If I took my liver and I put it in your body, you lost your liver, and I took a chunk of my liver and put it in your body, what would happen?
The liver wouldn't identify as the person that you gave it to.
God. Genius. Thank you for paying attention. That's right. Your body would say, this ain't me. This is not me. How does it know? That. All the stuff that's on the outside of the cell. All this stuff, all these proteins and lipids and glucose molecules sticking outside the cell is different for everybody. Are you listening to video?
Huh?
Are you listening to a video?
Yeah. Well, the other video from yesterday.
Baby, don't do that. You're going to get confused. Just pay attention because it's important, okay? This identifies you as you, okay? So this is what we call, your body would reject my liver. Why would it reject it? Because your body would say, this is not me. Sometimes your body gets confused because of an infection or something else. Maybe it's genetic. But your body gets confused and your body says, whoa, this isn't me, and it is you. And it starts attacking you start attacking yourself. We call those autoimmune diseases. So things like MS and lupus. Have you heard of lupus? And MS. Unfortunately, it's mostly more often women have MS and lupus than men. My brother had MS and he died of it, so men do get it, but women tend to get it more often. So it's important that you understand that autoimmune disease, an allergy like asthma, right? Asthma is also an autoimmune disease. Your body's reacting to this stuff that's on the outside of the cell. It gets confused. For some reason, it doesn't identify that as you, and you start having an autoimmune disease. You start to get constriction of the airways and all the other stuff that happens during an immune reaction. Okay? So that happens, but this identifies you as you, and one of the things that you can identify with is, one of the things that you've all heard of is blood type. And we'll be talking about that as we go on into genetics, but blood type in the United States, we have 3 major blood types. Well, actually there's more, but A, there's B, there's O, there's AB, are the 4 blood types. And you would call these, and maybe you don't, maybe you remember this word from middle school or elementary school, you would call this phenotypes. Do you remember that word, phenotypes and genotypes, from Punnett squares and all the stuff you did in middle school? So phenotypes, ABO blood groups are what we call phenotypes. Then there's a subgroup that, what is ABO? Well, A is one shape that's out here. B is another shape that's out here. O is another shape that's out here, or not, actually it's a not shape. And AB is both the A and the B shape that are out here together. And then there's another smaller one that we call, for blood, that we call plus or minus. So you're A positive or A negative, B positive, B negative.
I thought phenotypes was like characteristics. I thought it didn't have to do with anything like blood types.
That's really, really a great point. I love that you guys are on it today. Man, I don't know what happened, but please, let's continue this from now on. So yeah, that's the problem. That's the difference between middle school and high school. So you learn characteristics, that's right. What are characteristics? It's proteins and lipids and carbohydrates. That's what makes characteristics. What makes you tall? A protein made you tall, some hormone. What made you short? Some protein made you short, some hormone. What made you dark? A protein called melanin. What made you light? The lack of protein called melanin. Thank you. So what's this? Read the back. Oh, I'm so sorry. That's okay. I hope you feel better. You can lay your head down, dear. Does that make sense? So when we're talking about phenotypes, yeah, those, you're right. Characteristics are what we would call, there's a red. Is the flower red, or is it yellow, or is the plant tall or green? Is it wrinkled or smooth? Is it, do you have straight hair or curly hair? These are all characteristics, but they're all based on proteins. And when you're talking at the cellular level, then you're only talking about a single protein, and that's usually A, B, O in the case of blood types. Is everybody okay with that? Great questions, fantastic interaction, I love it. Now, let's go on and talk about some of the other organelles that we're going to need to know. Then we're going to get on to genetics. We're done with organelles, and we're going to deal with energy first. We'll do mitochondria and chloroplasts, and then we'll do endosymbiosis theory and photosynthesis and respiration, and then we'll go on in genetics. So before you go, I would like you to make sure you know that you need to know every organelle, including the nucleus. Notice I'm drawing the nucleus. You need to know the endoplasmic reticulum, the rough, which means that there are ribosomes on there. That's a ribosome is another organelle. Ribosomes do not have a membrane. So prokaryotes have ribosomes, and so do eukaryotes, but prokaryotes don't have an endoplasmic reticulum. So this is the smooth ER, smooth endoplasmic reticulum. This is the rough endoplasmic reticulum. You have a Golgi body. These are all membranes. These are all membranes. You have vesicles. You have mitochondria. All have membranes. And if you're a plant cell, you have chloroplasts. And chloroplasts, again, have membranes. All right, so know your organelles, know what they do, and we will get into mitochondria and chloroplasts tomorrow. There's a lot of organelles. These are the major ones. Let's go back and let me make sure I label these. So these here would be, this would be a vesicle. Obviously that's a smooth ER. The smooth ER, what happens is that vesicles leave, and they travel from there all the way to the Golgi body, and any vesicle that's traveling through the Golgi body has embedded in it proteins that were made, and it fuses. So to be clear, when it gets see how it's fusing? It becomes part of the Golgi body. Now remember, this is a three-dimensional object. I keep saying that, but this is not two-dimensional. This goes all the way down. This is it's a folded membrane. It's like a sac. So is this. Goes all the way around the cell. Goes all the way around the nucleus. The endoplasmic reticulum goes all the way around the nucleus. And these vesicles leave on the other end. They come out on the other end, and they go and they fuse with either the cell membrane. That's how you get proteins that are coded by the DNA out and go to the membrane, or they go and they join another organelle. But these are for proteins that are made in the cell that are going into a membrane. This is a mitochondria. Oh, I should label this as the Golgi body or Golgi apparatus. Always capitalize the G because it's someone's name. Mitochondria. And this is a chloroplast. Chloroplast cell wall. What kind of cell is this? Plant cells, correct. And one organelle that you don't really see here too well, it's a big membrane-bound organelle. I wish I could draw it differently somehow. Maybe I can. Let me try to do it with a pencil here. It might look different. Hopefully it does. Big organelle that kind of goes through the entire cell. Really is probably the biggest organelle in the plant cell. It's called a central vacuole. Anybody know what a central vacuole does? Nobody? Why don't we leave that for homework? The first person that brings it to me. Central vacuole. Tell me what it does, and I'll give you extra credit points. Whoever does it first. Is that fair? Central vacuole does something. I wonder if I could change it. I wish I could change the opacity. Anyways, the central vacuole is a really important part of the plant cell. So those are kind of the major organelles you're going to have to know. In the animal cell, you don't have the cell wall, but you have everything else that you see there except for the chloroplast. You have DNA, you have the endoplasmic reticulum, the Golgi body, the mitochondria, and there's a lot of them. You don't have a central vacuole. You do have vesicles. You do have a bunch of mitochondria. You do have a cytoskeleton. You do have a cell membrane. And it is a rough ER and a smooth ER. Ribosomes are in the cell, in the cytoplasm as well. By the way, that's another organelle. It's a cytoplasm. That's the liquid part of the cell. You have ribosomes, which make proteins. Those are the kind of the major organelles. Of course, you have things called lysosomes. It's our another little vesicles that are called lysosomes. So that should be a good review of all the organelles, everything you should know. Can you draw all this? Could you do this on your own? If you had an essay exam, if part of your test on the final was to sit down and draw all this and label it, could you do it? And tell me what each thing does, the function as well. That might be important. All right, have a good day.